right, cool. So let's see what people had to say. Why don't you look like your pet hamster? Um, because they have animal genes and we have human genes. Yes, but I do want to mention that they have just we're animals too because we're not like plants. We don't get to sit in the sun and photosynthesize, but we do definitely have different genes. So that stands. I'm not a hamster. Correct. It's not human. Correct. Because it's your pet. Yes. They have different genetic codes. You weren't created by one. They're not human, not human, genetic. Because we don't have animal genes. Well, I guess we don't have hamster genes because hamsters can't make humans because they're your pet hamster. So there's no genetics. It's not human. Don't have the same genes. Not human because I was created by humans. Because the genes from people make us, and it is not human. Exactly, guys. Not a hard question. The reason you don't look like your pet hamster is pretty simple. You get your genes. The genes are what make you from your mom and your dad. And your parents aren't hamsters. They're humans. Unless your parents are hamsters, which is insane. And you are probably the smartest hamster known to man. Because here you are doing virtual school. That being said, ladies and gentlemen, not to be funny, but I do want to mention this at this point because sometimes it gets sensitive for people. And I just please understand, I am not trying to get anybody upset. I know that all of our families come in different combinations. Maybe you have a step parent, maybe you have two step parents, maybe your mom and dad are still together, maybe you live with an aunt or an uncle or a grandparent. So please understand, regardless of who you consider to be your mom and your dad, because of how your family dynamic works, please understand that in biology, when we talk about mom and dad, we are talking about the people, the individuals who donated the genes to make the offspring. So we are talking about the biological parents, maybe not necessarily the parents that have stepped into that role if they are not your biological parents. So again, I am not trying to get anybody upset or thinking about anything one way or the other. I just want to make that distinction very clear because oftentimes I have kids like, well, I don't look anything like my mom, or I don't do it, and then I find out like there was somebody who was adopted, which is wonderful. But that being said, obviously you're not going to share genetic information with somebody if they have chosen to be your parent versus the person who donated biological material to the fertilized egg that eventually became you. Okay, anyway, so enough of the disclaimer. So moving on into the notes, ladies and gentlemen. So it's all in your genes. So I just want to be very clear about that. The genes that make you, pretty much are the genes that you got from mom, from dad. Okay. So what we see here is a picture of chromosomes, and these chromosomes, ladies and gentlemen, um, you get one from mom and one from dad, like quite literally, like literally. Let me get my annotations going. So this gene may have come from mom, right? I'm sorry, chromosome. This chromosome may have come from dad. That being said, though, I do want to point out that this section is where your actual genes are. So again, human beings get 23 pairs of chromosomes. You get one of the pair from mom, you get the other pair, kind of like you have 2 feet, right? You have two chromosomes for you have 23 chromosomes that you get from mom, 23 from dad. So you have 23 pairs of chromosomes. And those little tiny bands are just wrapped up little things of DNA that we call genes, and that's where your traits are carried. All right, mouse. There we go. All right, so we talked about Mendel last week. We know that he was the pea plant guy, and we know that over about 8 to 14 years, he studied pea plants because, again, they're really, really easy to breed and they have a variety of traits. Specifically, they have like 7 traits that he looked at. All right, so this is going to be the first one that you are going to be writing on, ladies and gentlemen. All right, so Mendel found out through his experiments that traits, these characteristics that we see in organisms, can either be dominant or recessive. We've talked about this already. In pea plants, for example, and this is going to go in your chart now for your notes, one example of a dominant trait is tallness, and an example of a recessive trait would be shortness. Um, Another example we saw were purple flowers versus white flowers. Purple flowers are dominant, white flowers are recessive. We also saw green versus yellow pea plants, peas themselves, I should say. Green, I think, was dominant. Yellow is recessive. And it doesn't really matter if we get these backwards or not. Just understand that the reason why we really like the pea plants is because all of their traits are bimodal, meaning that there's no real mixing, right? Either you're tall or you're short, either you're purple or you're yellow. They don't come out with like a medium sized plant, they don't come out with like a purpley white flower. Okay, they don't come out with like a greenish yellow pea. There's no in between. They're all bimodal, so it makes it kind of easy. Now we know that there were at least 4 other traits that the peas exhibited. We know that there was the pod shape. We know that the flowers were either axial or proximal. We know that there were wrinkle and round peas, and there was one other one that I always forget. So it doesn't really. I mean, I guess it matters. I just don't remember what the heck it was. There were other ones, but again, these were all either dominant or they were recessive. So, how does Mendel's work actually even help genetics? So he develops 2 laws. He develops the law of segregation. There was nothing to write really in the last right there. Yeah, no problem. You good? No, you're good. You're good. No worries about it. I'd rather you say something than not get the information you need. If I can get the mouse to work now, that'd be lovely. All right. So all of the stuff that he did with peas comes up with 2 laws of genetics, okay? The first is the law of segregation, and I know that this is a lot to write. So let's just be clear. There is really only for this first one, all you really need to do, ladies and gentlemen, is this first sentence. The 2 parts of a gene, pair or allele, that separate from each other in the formation of a sex cell. So if you are reproducing sexually, and not everything does, you will need to create a sex cell. So in females, we make eggs. In males, they make sperm. And if you are going to reproduce sexually, sperm and egg have to be introduced to each other and create a fertilized egg, or a zygote, which is the biological term for that. The law of segregation tells us that remember how we said that all genes come in pairs? Remember, you got one from mom, one from dad, all genes come in pairs. The law of segregation tells us that when I make a sex cell, when you make a sex cell, when everybody makes a sex cell, those pairs get separated again. So I can see that in this drawing. My pairs of chromosomes that would normally hang out together end up getting separated from each other. Okay, so that is the law of segregation. So, one more time, all of our chromosomes come in pairs. During the making of a sex cell, whether egg or sperm, depending on if you're male or female, those pairs become segregated. They become separated from each other. Mendel also came up with a second law, And that law was the law of independent assortment. So the law of independent assortment tells me that all of my jeans, and again, so like see here we see the two tall ones together, we see the two red short ones together, and then these are kind of like together in a grouping. We see these two tall ones together, we see these two short ones together, and they're together in a grouping. Well, look what happened to the short ones. Like there ended up being a red one came here, but then the other red one went over there. Okay, so the short red one's hanging out with the tall yellow one. And then all the yellows were hanging out together here, but now one of these little yellow ones is hanging out with the red, and one of these little yellow ones is still hanging out with the yellow. So what does this mean? This means that when you make a sex cell, all of your traits, not only do they separate and segregate those pairs, but then they also get shuffled. So literally it's like shuffling a deck of cards. So think about separating a deck of cards into two stacks, right? Maybe you separate your red cards from your black cards, but then you shuffle them and they can end up in any order whatsoever that you want. This is why we see so much variation when it comes to genetics, because literally there are hundreds of different combinations. I mean, this simple organism, which apparently only has 4 sets of chromosomes, can come up with, I don't even know what that number would be. I suppose I can do it mathematically. No, I can't because I don't have my good calculator with me. 4 times 3 is 12, times 2 is 12. This one cell could technically come up with 48 different combinations of those 8 little genes. So literally 48 different combinations. And that's just with 4 sets of chromosomes. Human beings have 23 sets of chromosomes. So that gives you an idea of why we see so much variation in human beings. Tall, short, really dark skin, really light skin, curly hair, wavy hair, straight hair, blue eyes, hazel eyes. All these different combinations are because of the law of segregation, the idea that these pairs of chromosomes separate, and then the law of independent assortment, the idea that when we make sex cells, everything's kind of gets shuffled out and they could randomly end up in any of the sex cells. Clear. Oh, excuse me. All right. So Mendel, we're going to watch a lovely little short video, hopefully, and this is going to introduce us to the idea of Punnett squares again, because I think my original introduction yesterday was an abject failure. These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century. The man is Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding from seed plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of genes. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square.
You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's keys first. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase Y always overpowers its lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous P mom will give each P kid a yellow dominant allele, and the green homozygous P dad will give a green recessive allele, so all the P kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited. But it all started with Mendel and his peas.
All right, so hopefully that was a quick little introduction to the Punnett squared, which hopefully made a little more sense than what we were talking about yesterday. Because, like I said, I got so many questions yesterday. I feel like my introduction didn't work at all for anybody. All right, so putting it all together, ladies and gentlemen, everything that you need to write will show up in red. All right, so Mendel found that his experiments that come in different crates could be either dominant or recessive. Remember, when we have a dominant gene present, that's the one we're always going to see, because dominant genes always kind of hide or mask the recessive gene. So, meaning, what this means is that the dominant trait, again, the red words are the ones that you're writing, is the one that showed up in the population more often, while the recessive gene trait hid or receded into the background. So again, it's kind of hiding, it's getting covered up, it's sort of being masked out by that dominant gene. Please don't put things in the chat, guys. I cannot see things in the chat when I'm sharing my screen. I see that there is a chat, but I can't see what's in the chat. So if you have a question, please make sure that you are actually unmuting and speaking up.
No, I was saying, can you please go back one, because I finished that one.
Yeah, the other one was literally just two words: the words dominant and then the word recessive. Okay, so ladies and gentlemen, again, so dominant traits always show up. Recessive traits tend to get hidden. All right. We call the dominant and recessive genes alleles. Now remember, you get one of these from mom, and you get the other one from dad. And depending on what combination you get of alleles will determine the traits that you have. Now, again, this is review. We know this. Dominant alleles are always represented with a capital letter. Now, we're using the letter B. It literally could be any letter, depending on what letters you're using, and that's okay. Oops, come on. Recessive alleles are always represented with a lowercase letter. So again, dominants, these are the ones that will mask the other alleles. These are always capital letters. Recessive alleles are always represented as a lowercase letter. Now, two of the same alleles. So let's say I get two that is the same. I got one from mom and it's identical to the one that I got from dad. We call these alleles then homozygous. Homo meaning same. Zygous is referring to a zygote, and a zygote is a fertilized egg. So what that means, the word homozygous literally means that the fertilized egg has two of the same alleles. Homo, same, zygous, fertilized egg. Now, another word that we could use instead of homozygous is purebred. So here we can see where I have two capital B's, homozygous, they're both the same, or I have two lowercase b's, These are homozygous or purebred because two lowercase b's are exactly the same. If I have two different alleles, in other words, maybe I got one from mom and I got something different from dad, then I have what we call a heterozygous or a hybrid offspring. Hetero means different. That word zygous, again, still refers to the zygote, which is the fertilized egg. So literally the word means the fertilized egg has something different in it. So heterozygous or hybrid, we have two different alleles. So maybe you got the dominant allele, the big B from mom. Maybe you got the recessive allele or the little b from dad. Okay? Really doesn't matter. Maybe you got the big one from dad or the little one from mom. It doesn't matter. You got one dominant from one parent, one recessive from the other parent. It makes you heterozygous or a hybrid. All right, so here are our possible combinations. If I have big B, then I am homozygous, dominant. Homozygous, purebred. Dominant, all I have are dominant genes. So whatever the dominant trait is, you're going to see. So let's say that this is for brown eyes because brown eyes is dominant over blue eyes. Homozygous dominant individual you would have brown eyes, and this individual only has brown genes or brown alleles. Another combination that we see is a heterozygous dominant. Okay, why is it heterozygous dominant? Because it's heterozygous. I have one big B, one little b. They're different, right? It is a hybrid. But remember, your dominant gene is always going to sort of mask or cover up that recessive gene. So I am still going to see the brown eyes on the outside of this person. But this individual carries a gene for blue eyes and they carry a gene for brown eyes. Our last possible combination is little b. And again, this is homozygous recessive. Homo, same, zygous, fertilized egg. So in my fertilized egg, I have two of the same genes. And both of those genes are recessive, meaning it is the one that the only see if they're both present. So in this case, if I have lowercase, or if I am homozygous recessive, then I would see the recessive trait. In this case, we've been talking about eye color. Specifically, you would be seeing blue eyes. Issues or questions so far? Like I said, most of this is review. Issues or questions going once? Issues. No, that's fine. What do you see? What do you need out?
It says could leave and then or, and I didn't see anything for the blank parts.
Read me this part again.
It's lower. Why it's taking so long?
You're fine.
Alright, in depth in his experiments, like the different traits could be blank,
dominant, or recessive.
Okay.
Yep, no problem. Any other issue? Yeah, you're welcome. Any other issues or questions? Okay, hearing none, we move on. Okay, so again, ladies and gentlemen, the letter combination of alleles is called the genotype. This is what you're actually getting from mom and dad. These are the genes that they are contributing to their offspring. Okay, so again, letter combination is your genotype. So if I'm talking about Capital T for dominant, tall in a pea plant. I'm talking about lowercase t, recessive is short in a pea plant. What are my possible genotypes? What could the genotypes be? Well, they could be a dominant genotype where I have capital T, or capital T, lowercase t. But remember, either of those combinations, whether it is homozygous dominant or heterozygous dominant, the plant would still look tall. Okay? The other possible combination would be little t. Again, this is homozygous, they're the same, but this is homozygous recessive because they're both carrying the recessive gene. In this case, ladies and gentlemen, what we would see, of course, is a short plant. So again, what the genotypes could be are, again, two capital T's, two lowercase t's, or a combination of those two, both little t and big T. The physical expression is what we call phenotype. The thing that I see on the outside. Is the plant tall? Is it short? Does the person have blue eyes or brown eyes? I mean, I can't look at you and see what's in your genetic code, obviously. I mean, duh. But I can't look at you and see your physical characteristics or traits. I can see if you're tall. I can see if you have blue eyes. I can see if you have a wide nose or a narrow nose. The physical expression of these genes is what we call phenotype. So, for example, if I see a dominant trait, what I would actually see for this point is a tall plant. So the phenotype, if I have capital T, or if I have capital T lowercase t, that's a dominant phenotype. I see tallness. However, if I'm looking at my recessive trait, what do I see for the recessive trait? I see shortness. So the recessive phenotype is short because that is what we see on the outside. So all of this culminates in this idea of a Punnett square, which we've looked at in the video. I tried to introduce yesterday. We talked a little bit about today in the Do Now. So let's actually see how these work, shall we? Before I move on, issues or questions? Okay. So let's talk a little bit about young rat love. Love, exciting and new. All right, so two rats fall in love and they want to start a family. The male rat has a genotype of capital A, lowercase a, and this is what we call a gouty in fur, and it's basically a black-brown mix. The female, who is his partner, has a genotype of AA, and this is a black phenotype. So what we want to do is we want to figure out what possible combinations of fur will their offspring have. In order to do a Punnett square, you need to know the genotype and the phenotype of the parents. As long as we know genotype and phenotype of the parents, we can build a Punnett square. So, Punnett squares are actually really, really useful genetic tools. Again, they will help us determine the possible outcomes of offspring. They will show us the possible genotype combinations. And if we know the genotype combinations, then we will know the possible phenotype possibilities. But you cannot predict exactly what a particular offspring is going to be. You can just say what the chances are that an offspring will be whatever the heck it is. All right. So, again, now you have this already on your sheet, and I just need to retype it because, or rewrite it because it's not on my sheet, but that is okay. So you already have this, oh, let me do blue for the male because he's male, and yay, gender stereotypes. All right, so male we know is a gaudy. Which is kind of like a brown-black combination, and he has this as his genotype. So I have the phenotype and the genotype for the male. For the female, which I'll do in pink, Her phenotype is black, and we know that black is recessive, A. All right, so how am I going to put this together? How am I going to figure out the phenotype and genotype for my offspring? All right, so the first thing you are going to do in a Punnett square is you are going to distribute. Oops, sorry, I didn't mean to do that. I'm just trying to move this thing out of the way. You are going to distribute the parents' genotypes. So again, what you need to do first is you need to have the dad on top. So use my genotype for the dad, and you're going to place it up top. So again, law of segregation. I need to separate them into different columns. So the dad's capital A followed by the lowercase a for dad. I then am going to place the mom's genotype on the side. Again, law of segregation says I am going to separate them. So one goes on top, one goes on bottom. Okay, are you with me so far? Any issues or questions so far on how to begin to...
I don't think so.
One more time?
No, I don't think so.
Okay, cool. All right, so how to actually use the thing. So now remember, law of segregation, we separate them first. So that's what we just did. Now we have the law of independent assortment. We have to get them shuffled. So how we shuffle in a Punnett square is literally, it's kind of like a drop-down menu. We are literally going to drop this A down into both of these boxes. We are going to drop dad's other A down into both of these boxes. So again, we separated first, and now we are sorting or independent assortment. We are then going to do the same thing with mom's genotype. Her A gets distributed across. So here is her first A and second A. Her other A also gets distributed across A and A. So now these are the four possible combinations. Again, I have 1, 2, 3, 4 possible combinations for my offspring. So now I want to take a look at that. So obviously, if I have capital A lowercase a, capital A lowercase a, I have 2 out of the 4 of my offspring, which are going to be A. So 2 out of 4 is equivalent to 50%. So half of my offspring, there's a 50% chance that my offspring is going to have this genetic combination, which means that they are going to have that agouti, I think I spelled that wrong, but that's okay, coat. Half of my children have a chance of, or half of these rat children have a chance of having that agouti, the black-brown combination of a coat. Now I have this combination, AAAA, all lowercase. I have 2 out of 4, or 50%, of my other offspring that have an AA combination, which means that these guys are going to have the phenotype black. All right? So what do we have here? When this male and this female get together, they have a 50% chance of making a black fur baby. You have a 50% chance of making the combo, the agouti baby. All right? That last page on your assignment, ladies and gentlemen, is like the worksheet. That is where I will spend most of my time grading this. So your 1 through 12 on the next page is the actual assignment, not the notes themselves. If you have any issues or questions, hang back. If you